टुडे इज लाइक इन नेक्स्ट 35 to 40 मिनट्स मैं आपको एक नया फील्ड दिखाता हूँ आज, राइट, जिसमें हम जो बॉडी के अंदर डीएनए होता है कैसे हम उसके साथ नैनो टेक्नोलॉजी कर सकते हैं, एंड हाउ वी कैन गिव द इंस्ट्रक्शंस टू द बायोलॉजिकल सिस्टम्स यूजिंग डीएनए एज अ लैंग्वेज, यूजिंग डीएनए एज अ कोड, राइट। और ये सब हम लोग करते हैं डे टू डे लाइफ में। आई विल शो यू हाउ सम रियल एग्जांपल्स, राइट। नाउ बिफोर वी गो इनटू द डिटेल्स, लेट मी स्टार्ट विद अ स्टोरी, राइट। अभी सुबह का टाइम है 10 बजे का, आधे लोगों ने ब्रेकफास्ट कर लिया होगा या आधे लोग अभी करेंगे, राइट। एंड इफ आई टेल यू कि माउथ इज नॉट द ओनली ऑर्गन by which we are eating the food, am I correct or wrong?
Yes, sir.
राइट, अगर मैं ये बोलता हूं कि हम नाक में से भी खाना खा रहे हैं तो ये स्टेटमेंट सही है? सही है, क्योंकि हम ऑक्सीजन जो है ये भी एक प्रकार का न्यूट्रिएंट है हमारे बॉडी के लिए। अगर मैं ये बोलूं कि हम आंखों से भी कुछ खा रहे हैं तो ये भी सही है, राइट। वी नीड सम लाइट काइंड ऑफ सिग्नल्स. If I tell you that we are eating through ears, that is also correct. If I tell you that we are eating through skin, that is also correct, right? The mechanical signals जो हैं वो बॉडी के अंदर जा रहे हैं जिसको बोलते हैं। इसी तरह से if we see our human body, it is made up of fundamental units called as cells, right? And जैसे हमको न्यूट्रिएंट्स चाहिए, हमको ऑक्सीजन चाहिए, उसी तरह से सेल्स को भी न्यूट्रिएंट्स चाहिए होते हैं for all the physiological purposes. Now, all this started in 1940-50 when the process called as endocytosis, the process by which the food or the nutrient, not only nutrient, it can be toxins also, some harmful chemicals also, or drugs, how they go inside the body was a big challenge of the research. And people discovered this protein, अगर आप number 1 देख रहे हैं स्क्रीन पे, this is called as clathrin mediated endocytosis, जो हमारे टंग की तरह है, राइट, कि जब सामने खाना आता है तो हम जीभ बाहर निकाल के खाने को अंदर ले लेते हैं। Exactly, that's the way by which this protein called as clathrin works, कि जैसे ही nutrients from outside, the moment they come and sit on the plasma membrane, when they bind to the receptors, this protein forms a cavity around the piece of plasma membrane. And जो ऑरेंज कलर का प्रोटीन देख रहे हैं इसको डायनामिन बोलते हैं, that cuts the vesicle and the molecules go inside. Now this was the hypothesis known for say 40 to 50 years, and around 1985 or 90, 2 new pathways got discovered. One is called as small interfering RNA, siRNA, which got the Nobel Prize this year in medicine and physiology. And second was the small molecule based inhibitors which could block any functional protein, कि जो आज हमारे पास available हैं, like using small molecules you can block the function of a protein, any protein in the cells. तो जब ये प्रोसेस को बंद किया गया क्लैथरिन मीडिएटेड पाथवे को, तो ऐसा देखा गया, जस्ट 1 सेकेंड भैया मैं मीटिंग में आया। सो जब ये क्लैथरिन को ब्लॉक किया गया तो ये देखा गया कि द न्यूट्रिएंट्स आर गेटिंग एंडोसाइटोस्ड even at an uncompromised rate, कि 90% of the molecules are going inside the 90% of the molecules are going inside the cell without any uncompromised efficiency. And that gave rise to the concept that there are alternative pathways available for the endocytosis, right? कि जिससे molecules अंदर जा सकते हैं. And that led to the discovery of many pathways like the caveolin, bar domain mediated proteins. लेकिन क्या हुआ था till 2005 or 6, only pathways which were discovered were the ones which are mediated from inside the cell, कि क्लैथरिन है, कैवियोलिन है, बार डोमेन्स हैं, ये सब अंदर से रेगुलेट करते हैं. Now I'll again take one example. आपने अभी सबने नाश्ता किया होगा, everybody has their stomach full. But if I show you again 2 very good gulab jamuns और आइसक्रीम और जिलेबी outside, right, आपका जी लग जाएगा कि शायद थोड़ा और खा लेता हूँ, भले हम अंदर से फुल हैं, but this time the force or the push has come from outside, correct? उसी तरह से अगर आप कैंसर प्रोग्रेशन देखते हैं, जिसे एपिथेलियल टू मेसेनकाइमल ट्रांजिशन कहा जाता है, the pathway, of course the cancer is in the nucleus because of the changes in the genetics. There are a lot of factors which are affecting the pathways from outside the cells, जैसे कि मैकेनोबायोलॉजिकल सिग्नलिंग होता है, right? या केमिकल सिग्नलिंग होता है फ्रॉम आउटसाइड द सेल्स, एंड दैट इज आल्सो एप्लीकेबल टू स्टेम सेल्स डिफरेंशिएशन. जब हमारा बर्थ होता है, हाउ डू द स्टेम सेल्स आर गेटिंग डिफरेंशिएटेड, अ लॉट ऑफ दोस प्रोसेसेस or physiology is actually regulated by the external signals, not the internal. And then this question was asked that are there processes from outside, or are there proteins from the extracellular matrix which can dictate the endocytic pathway? And उसमें भी तीन चार pathways हैं, and one pathway which was discovered by my postdoc lab, Professor Ludger Johannes in Paris, was कि कुछ प्रोटीन्स होते हैं, a class of proteins called as galactin. These are called as lectins, which means they have affinity for carbohydrates, the sugars. These proteins are secreted from the endoplasmic reticulum to the outside of the cell, and they can bind to glycosylation. Now remember, अगर आप लोगों ने थोड़ा बहुत प्लाज्मा मेम्ब्रेन या बेसिक बायोकेमिस्ट्री पढ़ा होगा, तो जितने भी प्लाज्मा मेम्ब्रेन के प्रोटीन्स हैं, जैसे कि ग्रोथ फैक्टर रिसेप्टर्स, CD44, राइट, इंटीग्रिन्स, ऑल दिस प्रोटीन्स आर ग्लाइकोसिलेटेड, जिसको बोलते हैं पोस्ट ट्रांसलेशनल मॉडिफिकेशंस. कि जब प्रोटीन्स एंडोप्लाज्मिक रेटिकुलम में बनाए जाते हैं, दे कम टू गोल्जी एपरेटस, उस पे उनका मॉडिफिकेशंस होता है, जिसको PTMs बोलते हैं या पोस्ट ट्रांसलेशनल मॉडिफिकेशंस बोलते हैं. एंड देन these proteins are secreted outside. The moment these proteins are secreted outside, they start to bind the receptors on the plasma membrane, and then these proteins have the capacity to induce membrane bending and pinching, forming of the endocytic vesicles without clathrin requirement or without any internal protein requirement, right? This gave rise to the new pathway where the fate of the endocytosis is actually regulated by outside, not inside, right? Now there were a lot of questions ki, which proteins are going through this pathway, in which cell types this is operational, does it changes in the cancer or does it changes in the stem cells, all these questions were there. अब उसको कैसे पढ़ाई किया जाए? अगर मैं बायोकेमिस्ट्री या मॉलिक्यूलर बायोलॉजी के स्टूडेंट्स को ये सवाल पूछता हूं कि अगर मुझे किसी प्रोटीन का फंक्शन स्टडी करना है, तो वो लोग सब एक ही जवाब देंगे, tag the protein, correct? Tag the protein with GFP or his tag or any other tag, and then study. Problem is this, these are very small proteins. Remember, green fluorescent protein GFP itself is 28 kilodalton, and these proteins are all 20 to 25 kilodalton. तो अगर एक छोटे प्रोटीन के ऊपर बड़ा टैग लगाऊंगा तो वो भी मुसीबत है. एंड these proteins are oligomerizing, राइट? सो उसमें भी प्रॉब्लम है. तो हमें एक ऐसा टूल चाहिए था जिससे हम सिंगल प्रोटीन का स्ट्रक्चर और फंक्शन स्टडी कर सकें without modulating its structure. And then what we said is, चलो कोई nanotechnology का टूल ढूंढते हैं जिससे, let us say a gold nanoparticle, a simple gold nanoparticle अगर मैं टैग करता हूँ प्रोटीन को, तो using electron microscopy I can study the organization of the proteins, correct? अगर मैं फ्लोरोसेंट क्वांटम डॉट यूज करता हूँ, तो मैं सिंगल मॉलिक्यूल माइक्रोस्कोपी से उसका स्टडी कर सकता हूँ. या तो फिर अगर कोई मैग्नेटिक नैनो पार्टिकल है, तो मैं इन एंडोसिटिक वेसिकल्स को प्यूरिफाई कर सकता हूँ by magnetic purification, and then I can study by mass spectrometry, proteomics, correct, which proteins are going through this. So इसको लेके हमने शुरू किया था रिसर्च 2007 में, and all this started in India by a professor called Yamuna Krishnan, who was a PhD student from IIC Bangalore, postdoc at Cambridge University, and then she came to TIFR, Tata Institute of Fundamental Research, and I happened to be her second PhD student from Bangalore. And we are all chemistry people, we are all chemistry students, but we look at the molecules, biological molecules from chemist eye, right? कि जैसे आप सब लोगों ने बेसिक पढ़ा होगा, डीएनए का स्ट्रक्चर पढ़ा होगा, प्रोटीन का स्ट्रक्चर पढ़ा होगा, राइट? अभी मैं आपको एक एग्जांपल पूछता हूँ सभी स्टूडेंट्स को, ठीक है? आप सब लोगों ने ड्रोन्स चलाए हैं, राइट? आप सब लोगों ने मोटर्स यूज किए हैं, राइट? क्या कोई बता सकता है कि बायोलॉजी में सबसे पहला नैनो मशीन कौन सा बनाया गया था? कोई स्टूडेंट आंसर करना चाहेगा? व्हिच वाज द फर्स्ट नैनो मशीन ऑटोमेटिक ड्रिवन? हम लोग एक्टिवा चलाते हैं, राइट? कार चलाते हैं, सब मशीन्स में ही है. कोई बता सकता है पहला मशीन कौन सा नेचर ने बनाया था अंदर? हेलो, am I audible, sir?
Yes sir, you are audible, very much audible.
राइट. तो चलो मैं आंसर बताता हूँ आपको. डीएनए पॉलीमरेज. अगर किसी ने पढ़ा है बायोकेमिस्ट्री में, दिस इज द एंजाइम which can replicate, which can make copies of the DNA, correct? DNA polymerase, ये जो machines हैं, and look at those enzymes or look at those assembly from a nanotechnologies point of view, what do you realize कि ये प्रोटीन्स नहीं है, ये एक नैनो मशीन्स हैं कि जो इनपुट लेके डेटा को प्रोसेस करते हैं, उसको आउटपुट देते हैं, करेक्ट? तो अगर डीएनए पॉलीमरेज सेल्स के अंदर ये सब कर सकता है, तो क्या मैं उसको टेस्ट ट्यूब के अंदर नहीं कर सकता? टुडे एवरीबडी इज डूइंग दैट. अगर आप बीएससी के प्रैक्टिकल देखेंगे, एमएससी के प्रैक्टिकल देखेंगे, वी डू समथिंग कॉल्ड एज इन विट्रो ट्रांसक्रिप्शन, वी डू इन विट्रो ट्रांसलेशन, करेक्ट? उसी तरह से नाउ look at this molecule DNA as a polymer. Don't look at it as a genetic material. Of course it is genetic material, it can store the information, लेकिन इसकी बहुत अच्छी खासियत है. What are the two खासियत of the DNA? One is जो single stranded DNA होता है, अगर उसका कोई sequence है, it will go and bind only to the other sequence which is fully complementary to it, जिसका stability सबसे ज्यादा हो, thanks to the Watson-Crick base pairing. सेकेंड, आई कैन फोल्ड द डीएनए. जैसे कि न्यूक्लियस के अंदर डीएनए फोल्ड होता है, उसी तरह से हम टेस्ट ट्यूब में भी डीएनए को फोल्ड कर सकते हैं. एंड लुक एट दिस एग्जांपल्स. दीज आर ऑल द एग्जांपल्स ऑफ द नैनो डिवाइसेस व्हिच आर मेड अप ऑफ द डीएनए. यू कैन फोल्ड द डीएनए इन अ टेस्ट ट्यूब. And then even you can do something called as computation, जिसे हम बोलते हैं डीएनए बेस्ड रोबोटिक्स और डीएनए बेस्ड बायोसेंसर्स. अभी 5 साल के बाद हमारे पास ऐसे मोबाइल फोन्स आएंगे जिसमें डीएनए चिप्स होंगे. अभी आप देख रहे होंगे, राइट, वी हैव 128 GB स्पेस, 256 GB स्पेस. अगर मैं ऐसा मोबाइल आपको बना के दूं जिसमें 100 टेराबाइट स्पेस होगा, राइट, नाउ विथ सिलिकॉन चिप्स इट इज इम्पॉसिबल. लाइक ऑफ कोर्स इट विल बी पॉसिबल, बट बहुत चैलेंजिंग होगा. Imagine if we could have a technology by which we could store this information in the DNA, right? I can make trillions of terabyte of the space using even a small amount of DNA. This is called as DNA computation. And all these are, just one second. Babu, Ritu isko leke ja, Babu. Sorry. So ये है मेरा lab. Like this is the overview of my lab. What we do in my lab is we fold the small pieces of DNA into three-dimensional. Just one second. Sorry for the disturbance. जी. So यहाँ पे हम देखेंगे. In my lab, we fold the DNA into 3D nano cages. Like using technology called as DNA origami, we can fold even the genomic DNA or small pieces of DNA into any shape that we want using computation-based softwares. And now हम लोग artificial intelligence और machine learning भी use कर रहे हैं, जिससे आप कोई भी shape बना सकते हैं, right? The question is, why do we want that? Like अगर आप देखेंगे तो इसके अंदर एक बहुत अच्छी स्पेस है. All these cages have a very nice cavity, which means you can put some molecules inside it. चाहे वो नैनो पार्टिकल हो, चाहे प्रोटीन हो, क्रिस्पार हो, एंटीबॉडी हो, एंड देन उनका एक बहुत अच्छा सरफेस है, 3-dimensional सरफेस, जिसको मैं कपल कर सकता हूँ. I can couple it to the antibody, I can couple it to the aptamer, and then I can target it to any cell in my body or any tissue in my body for the targeted release of the drugs, right? So this is the overview of our laboratory. We make different DNA nanostructures. We do a lot of functionalization in terms of chemistry, and then we apply them for biological imaging, sensing, drug delivery, right? एक example मैं आपको दिखाता हूँ इसका, जो मेरा PhD का काम था, right? If you see viruses, right? आप सबने viruses के बारे में पढ़ा होगा. These are the protein-based nano cages which can encapsulate a nucleic acid, correct? COVID, RNA, adenoviruses, all those things you have studied. And if you see the geometry of the viruses, most viruses have icosahedral geometry, correct? आपने देखा होगा जो 3 proteins होते हैं, they polymerize into an icosahedron. अगर मैं आपको पूछूं कि सिर्फ icosahedron ही क्यों? Why not a cube? Why not a buckyball? Why not a octahedron? Right? Nature ने सिर्फ icosahedron क्यों बनाया viruses को? तो इसके जो आंसर्स हैं, अगर आप ज्योमेट्री प्रिंसिपल्स देखेंगे, प्लेटोनिक सॉलिड्स, icosahedron, out of all the platonic solids or the archimedean solids, just give me 1 second, sir. Yeah, right. Just excuse me for only 1 second, please.
If any of you have any question, you can write in chat box or you can ask to him.
हाँ, anytime. I just need 10 seconds. Only just 10 seconds.
Yes. आप सब करिए. And if you have not joined my LinkedIn our YouTube. Then you can join my LinkedIn, YouTube also, to keep in touch and attend next lecture also.
yes sir, आई एम बैक हियर जी जी. हाँ, सो अगर आप देखेंगे वायरसेस को, राइट, नेचर ने वायरसेस को आइकोसाहेड्रल सिर्फ इसलिए बनाया था क्योंकि आउट ऑफ ऑल द प्लेटोनिक सॉलिड्स, आइकोसाहेड्रॉन इज द वन व्हिच हैज द मैक्सिमम एनकैप्सुलेशन वॉल्यूम एंड मिनिमम पोर साइज. इसीलिए जब उसके अंदर आरएनए वैक्सीन या डीएनए एनकैप्सुलेट हो जाता है, दे विल नॉट कम आउट. And based on the same principles, inspired from the viruses, what we did is we used small pieces of DNA. अगर आप ये small pieces of DNA देखते हैं, ये these are primers जो आप लोग सब खरीदते हैं, right, PCR के लिए. उन्हीं primers को हमने सिर्फ ऐसे modify किया कि they will now start to self assemble into a full virus, a icosahedral virus. और जब ये मशीन में हो रहा है, तब हमने क्या किया था, उसमें नैनो पार्टिकल्स डाल दिए थे, फ्यू गोल्ड नैनो पार्टिकल्स. सो नाउ इफ यू प्यूरिफाई दिस बाय द HPLC टेक्निक एंड विजुअलाइज बाय द इलेक्ट्रॉन माइक्रोस्कोप, यू विल सी that here are the nano cages. White color जो है ये DNA है by uranyl acetate staining, and these are the gold nanoparticles inside this. And I told you कि DNA का जो surface है उसको हम functionalize भी कर सकते हैं. So we added a small molecule called as folic acid. We attached a folic acid, and remember, out of all the cells in our body, cancer cells have a very high expression of a receptor called as folate receptor. So if you now take the cancerous cells, these are IA2.2 cells, and इसके ऊपर अगर हम ये DNA cages डालते हैं, we saw that they are nicely getting inside the cancerous cells. This told us that not only am I going to encapsulate the molecules, I can even modify the surface of the DNA cage for specific targeting, correct? एंड यहाँ पे हमने ये जो एक छोटा सा मूवी दिख रहा होगा, we used super computer in IISc Bangalore and we attached these molecules at different sites to find out कि ऐसा कौन सा पोजीशन है जहाँ पे ligand जो है वो बाहर की तरफ फेस कर रहा है, so that when you put these molecules on the cell surface they can interact with the receptors, correct? ये स्टडी था हमारा. Now, can we apply them in the live cells and animals? Answer is yes, right? I will give you one example. If you see the cancer chemotherapy, right, हमने कभी ना कभी अपने लाइफ में देखा होगा किसी को कैंसर की कीमोथेरेपी लेते हुए. One of the biggest problem even today with the chemotherapy is that out of 100 molecules that we inject in the patient, only a small fraction of the molecules reach the cancer, right? बाकी जो molecule है वो blood में diffuse होते रहता है, and the drug cannot distinguish between the normal cell and the cancerous cell, right? इसीलिए आप देखते होंगे कि जो patients कैंसर कीमोथेरेपी लेते हैं, किसी का बाल चले जाता है, किसी का आईब्रोज चले जाता है, किसी का किडनी फंक्शन में प्रॉब्लम होता है. So the bigger challenge was, can we develop a technology by which we can minimize the diffusion or the site targeting of the cancer drug and target it specifically to the cancerous cells? अब देखिए उसमें हमने कैसे किया. We took the free drug, for example dextran. This is a fluorescently labeled dextran, and we injected in an animal called as C. elegans. This is a small nematode which you can see by the microscopy, and we found that the drug is diffusing all over the body of the animal, जो आप देख रहे हैं ऊपर के केज में. Now what we did is we took the same concentration of the drug, put them inside a DNA cage, and then you inject it in the animal. जो आप lower panel में देख रहे हैं, you will see that the diffusion is almost minimum in the body, and it is targeted only to those cells where the receptors for the negatively charged molecules like DNA are getting expressed, right? So just one second please, एक second.
संडे सर का भी फैमिली टाइम रह रहा है.
नहीं सॉरी सर, थोड़ा वो there are some guests at the home, तो इसलिए थोड़ा सा ये सॉरी.
यस सर.
जी सॉरी. Is it visible sir slides? Yeah. हाँ जी हाँ. So यहाँ पे हमने first time we showed that with DNA nanotechnology we can remove the off-targeting effects of the drugs and target only to those cells where the receptors for the negatively charged molecules like DNA are applicable, right? ये सबसे बड़ा एक breakthrough था, and this has been patented now, and we are even trying to see if we can use them for overcoming the biological barriers like the blood-brain barrier. I'll come to it in a few minutes, right? So with this background, I will come back to the first question, where we were interested in understanding, can we functionalize the DNA cages with proteins like galactin 3 which can bind to the receptors, right? So यहाँ पे हमने क्या किया, we engineered the protein so that there is only one cysteine residue for coupling to the DNA, and then we encapsulated only one nanoparticle. We engineered the cage in such a way that only one nanoparticle could be encapsulated. ये आप नीचे देख रहे हैं electron microscope में, that there is only one nanoparticle and surrounded by one cage, and they are all monofunctionalized with the protein. So we used something called as co-localization studies on the electron microscopy, where there was a nanoparticle with antibody against galactin 3. And you can see there is one big, one small nanoparticle next to each other, indicating that all the nanoparticles are monofunctionalized, right? So ये पहली बार हमने दिखाया था कि with DNA nanotechnology you can control the number of ligands on the nanoparticles, right? Now the bigger question was कि चलो protein तो couple कर लिया, but is it really functional? Does the protein works in the cells, right? तो उसके लिए हमने electron microscope use किया. यहाँ पे तो आप देख रहे हैं, this is called as cryo super resolution scanning EM, not the TEM. Scanning electron microscope में cryo conditions में आप cells को भी देख सकते हैं. So these are the HeLa cells that you are seeing, and if you look at a very small piece of the plasma membrane, you will see that these are the DNA nano cages which are binding to the receptors on the cell surface. Now this was done at 4 degrees where the cages are binding to the receptors. If you do the same experiment at 37 degrees, you see that they start to form the clusters, जो मैंने आपको कार्टून दिखाया था, and these clusters now can start to go inside the cells by process called as endocytosis, right? अब वो कैसे देखेंगे? Again for that we went into the cryo electron microscopy using the sectioning. तो आप cells को cut कर सकते हैं, and using electron microscopy you can see, जो ये नीचे का image आप देख रहे हैं, these are the endocytic vesicles, and अगर आप बस contrast change करेंगे तो आप देखेंगे कि all these endocytic vesicles are filled with gold nanoparticles, indicating that with DNA nanotechnology we can functionalize the protein without affecting its function. This was the first report where we showed using electron microscopy and super resolution imaging that you can target any protein with DNA nanotechnology, right? Now this opens a very big window now for high throughput research. And then what we did is we changed gold nanoparticles to quantum dots. Quantum dots are fluorescent nanoparticles, and they are very highly fluorescent, and that is the advantage that you can even track the single molecule or the individual protein molecules in the cells. तो हमने क्या किया, we took the fluorescently labeled galactin 3 and mixed it with the DNA cage carrying quantum dot and the galactin 3. And if you do the binding and the uptake studies, we see that these cages are going exactly to the location where the normal protein should be going. कोई mistargeting नहीं हो रहा है, right? They don't go to the nucleus or Golgi and all those things. Now, as I said, the advantage of quantum dots is that you can image the proteins at a single molecule resolution. So here is what you see. एक छोटा सा मूवी आपको दिख रहा होगा. This is called as TIRF, Total Internal Reflection Microscope, where we are looking at a small piece of plasma membrane where these molecules bind to the receptors and they start to diffuse to see the correct environment for their endocytosis. And the moment that happens, they get uptaken in a clathrin independent process. यहाँ पे जो मूवी देख रहे हैं, यहाँ पे microtubules जो हैं, they are labeled with GFP, and यहाँ पे एक छोटा सा, you can see a single bunch of microtubule where the quantum dots are actively migrating. यहाँ पे नीचे आपको co-localization भी दिख रहा होगा, like they move along the microtubule tracks, जो कि retrograde transport का एकदम characteristic है. And that told us that first time we can target a specific endocytic pathway using the DNA nanotechnology, right? And now with the artificial intelligence, machine learning, right? अब क्या हो रहा है? Quality and quantity both are getting important. कि अगर मैं ये बोलता हूँ कि clathrin independent pathway है, तो लोग ये पूछे how much it is, how much of the fraction is going through this pathway, that pathway. तो उसके लिए हमने यूज किया था एक नया टेक्निक कॉल्ड एज लैटिस लाइट शीट माइक्रोस्कोपी. If you remember 2014, Eric Bedzik got the Nobel Prize for super resolution microscopy. उनके ही लैब में ये माइक्रोस्कोपी बनाया है जिसको बोलते हैं लैटिस लाइट शीट माइक्रोस्कोपी, जहां पे नॉट ओनली द सेल्स, यू कैन इमेज द एंटायर टिश्यू, एंटायर एनिमल्स at the quantitative level, and you can see how much fractions of the proteins are going through that, right? India का first ऐसा मशीन जो है लैटिस लाइट शीट हैज recently been installed in CCMB, Center for Cellular and Molecular Biology, हैदराबाद, जहां पे आप पूरे सेल्स को, एनिमल्स को एक साथ देख सकते हैं, एंड वी कैन इवन डू द क्वांटिफिकेशन ऑफ दोस प्रोटीन्स, राइट? तो ये एक मूवी है जो हार्वर्ड यूनिवर्सिटी में लिया गया था. हमने सैंपल्स वहां पे भेजे थे, एंड माय फ्रेंड सेंथिल अरुमगम एंड श्री गोकुल, like they did the imaging, and you can see in 3D like how these quantum dots are going inside the cells, and we can even do a lot of quantification. So all that is just been accepted very recently. It got accepted in a journal called as Nature Cell Biology. It will be published in few days, and आप देख सकते हैं, one of the very prestigious journal in the community. And with this I came back to India, and when I came back to India, हमने थोड़े चेंज किए थे क्वेश्चंस. वी सेड that before even we ask the questions of drug delivery or immune modulation or stem cells, let us ask how are these molecules interacting with the cells, and by which pathway are they going inside the cells. So this was all the work done by my first PhD student Anjali Rajwar. Right now she is a postdoc in Harvard Medical School in Boston. उन्होंने ये PhD काम किया था, and hats off to her. Single-handedly she set up the lab also, did this work, and all in the COVID time. पूरा कोरोना के टाइम पे जब लॉकडाउन था, उसमें भी उस स्टूडेंट ने बहुत मेहनत किया है. So what we did is this. We said that can we make first the DNA cages of different geometries, that which geometry is optimum for the cellular uptake studies and in vivo applications.
सर एक क्वेश्चन है, लेंगे एक
हाँ
क्वेश्चन.
जी, बोलिए बोलिए. हाँ जी.
ज्योति का प्लीज आस्क
Yes,
क्वेश्चन.
sir.
Yes,
Yes, sir.
please introduce and then ask
Hello,
question.
sir. I am Jyotika. Sir,
हाँ
you
जी.
told that the target of or the interest of the DNA molecule is coated with the gold particle. So my
हाँ जी.
question is, when we insert that nanoparticle along with the gold particles, what the DNA is taken up by the expression system in the cell. Sir, what is the fate of that gold particle? Is there any
Very nice.
technique which is used
जी.
to detect the fate of that gold particles
Right.
inside the cell?
राइट. सो प्रॉब्लम विद द गोल्ड नैनो पार्टिकल इज आप उसको देख नहीं पाते सेल्स के अंदर, राइट? हमने जो दिखाया था यूजिंग द, अगर आप वो नेचर नैनोटेक देखेंगे, दे डेफिनेटली गो टू द लाइसोसोम्स, राइट, जो एसिडिक होते हैं, एंड इफ यू इमेज देम फॉर अ लॉन्गर पीरियड ऑफ टाइम, दे सम ऑफ द फ्रैक्शन of these gold nanoparticles are recycled back, like वो सेल्स के बाहर फेंक दिए जाते हैं, and some of them they go to lysosomes. Now of course lysosome में जाके वो डिग्रेड ही हो जाएगा, but the problem is gold nanoparticles are not fluorescent, so you can't see them getting degraded over time, right? तो इसीलिए हम लोग क्या करते हैं, we label the DNA instead of the gold nanoparticle, because DNA के ऊपर आप fluorophore लगा सकते हो, and we monitor the fluorescence intensity over time. एंड अप टू 48 days, 48 hours दे आर स्टेबल. 2 दिन तक वो स्टेबल होते हैं. उसके बाद क्या होता है, इवन एनिमल्स स्टार्ट टू डाई, तो एनिमल्स या सेल्स को भी उतने लॉन्ग टाइम के बाद इमेज नहीं कर सकते. तो वी डोंट नो वेदर द शेल्फ लाइफ इज बियॉन्ड 2 days और आफ्टर दैट दे स्टार्ट डिग्रेडिंग. But 2 days or 48 hours is actually optimum for drug delivery applications, and rather we want the DNA cages to be cleared off after the 2 days.
Please continue. Yes.
Does it answer, Jyotika?
Yes, sir. Thank you. But I have a very small question, sir. What will happen after DNA get inserted inside the cell? What will happen to that gold particle?
DNA will not insert. DNA will not be inserted in the cell. It goes to the lysosome, and lysosomes में वो degrade हो जाता है. So what we believe is even gold nanoparticles will be degraded over time, or they will be thrown out of the cell.
Okay, sir. Thank you.
Right. So what we did is we made DNA cages. Sir, slides दिख रहे हैं ना, sir?
Yes, sir. Yes, sir. दिख रहे हैं.
Okay. So we made DNA cages of different geometries to see how are they interacting with the cellular surfaces. And here पे हमने tetrahedron, cube, icosa and buckyball बनाया. As you, if you remember, we started the same question with the icosahedral geometry of the viruses, and then this is a very high resolution AFM studies जो आप देख रहे हैं. And then we started to ask the questions, what is the size and the geometry of the molecules that is optimum for the cellular internalization, right? So we made a DNA cage of different sizes, like 8 nanometers, 12 nanometers, 40 nanometers, and then we started to put them in the cancerous cells. These are MDMB 231 cells, and what we find is that there is size regime in which the molecules have highest uptake, right? So size हमें पता चला था, and then what we did is, keeping that size constant, we changed the geometry. And then what we did is you made a cube, buckyball, icosahedron, keeping the fluorophores constant, right? And यहाँ पे एक प्रोटीन है, this is called as transferrin, जो मैंने आपको शुरू में दिखाया था, clathrin marker. This is the internal control, that one protein is constant and then we change the another one, जिससे हम ratiometric uptake देख सकते हैं. And what we found was only tetrahedron was the one which showed the maximum uptake in the cells as compared to the other ones, right? We also did studies on different cell types from the body, and we found that the breast, liver and the kidney cells have the maximum affinity for the DNA cages. Like उनके ऊपर जो receptors हैं, they are positively charged, so they have affinity for negatively charged molecules like the DNA cages. अब इसमें एक बात देखिए, brain नहीं है. Targeting brain is very difficult, and I will show you in few minutes how we can do that, right? So वो जब था वो जब study हुआ, then we said, okay, what are the mechanisms by which the molecules are getting internalized? Now, if you remember my very first slide, there are different mechanisms, clathrin mediated pathway, independent pathway, और उनके लिए आज हमारे पास small molecule inhibitors available हैं, right? So what we did is we blocked a clathrin pathway using this molecule called as pit stop. अगर आप देखेंगे, the cells which are treated with the pit stop, transferrin endocytosis पूरा block हो गया, DNA cage uptake block हो गया, पर जो clathrin independent pathway है वो block नहीं हुआ, right? Which tells us that these molecules are going through the clathrin mediated endocytosis. Same was with the dinosaur, which blocks a protein called as dynamin, right? अब opposite देखिए, if I block the clathrin independent pathway by lactose, so lactose is a competitor for galactin, कि वो galactin को bind ही नहीं करने देगा receptors में. ये देखिए, the galactin pathway is completely blocked, transferrin is not affected, and DNA is also not affected, indicating that these molecules are going very, very specifically through the receptor mediated endocytosis and the clathrin mediated pathway, right? Now, ये जब होता है, आप एक बात याद रखिए, why are cancer cells so aggressive as compared to the body cells? Because in cancer cells, when the endocytosis is enhanced, along with the endocytosis, a lot of signaling pathways are also getting affected, right? आप लोगों ने थोड़ा बहुत पढ़ा होगा इसके बारे में, MAP kinase, SARC kinase, JAK-STAT signaling, and that is why the proliferation rate in cancer is also very high. So we said, can we test that model? So what you can do is, instead of growing the cells in 2D, you can grow the cells in 3D. जैसा कि बॉडी के अंदर एनवायरमेंट होता है. सो इफ यू ग्रो द MDMB 231 कैंसर सेल्स इन अ 3D एनवायरमेंट, दे फॉर्म समथिंग कॉल्ड एज स्फीरॉइड, जिसको सिंपल लैंग्वेज में बेनिंग ट्यूमर बोला जाता है. दिस इज द बेनिंग ट्यूमर दैट यू सी. एंड देन इफ यू एक्टिवेट दिस मॉलिक्यूल्स बाय द लिगेंड्स लाइक द DNA केज, दिस सेल्स स्टार्ट टू कम आउट, जिसको इन्वेजन बोलते हैं, और बेनाइन टू मेटास्टेसिस चेंज बोलते हैं, राइट? And again, what we did is we used this process of cellular invasion as a function of DNA geometry, and we found that the tetrahedron again had the maximum efficiency in inducing this, right? अब आप बोलोगे अगर ये बात है तो फिर तो कैंसर के लिए यूज ही नहीं होगा. Yes, it will not be used for the cancer because it is actually activating the signaling pathways. But where can we use them? अगर ये देखेंगे, neuroscience, the neurodegenerative diseases like the Alzheimer's or the Parkinson's, वहां पे क्या होता है, जो न्यूरॉन्स हैं उनका एफिशिएंसी दिन ब दिन कम होता शुरू हो जाता है. सो अगर उन डिजीजेस में, न्यूरोडिजेनरेटिव डिजीजेस में हम ये टेक्नोलॉजी यूज करते हैं, दे कैन एक्चुअली एक्टिवेट द न्यूरोनल सिग्नलिंग एंड द न्यूरोनल ग्रोथ, एंड दैट gave us a clue that okay, you cannot use it for the cancer treatment, but definitely you can use it for the neurodegenerative diseases like the Alzheimer's and the Parkinson's. But उसका एक problem है, getting these molecules to the brain is a big challenge, right? Because there is something called as a blood brain barrier. So what we did is we said, can we test this? तो उसके लिए एक एनिमल मॉडल आता है zebra fish. So here the skin of the zebra fish is labeled with GFP, green fluorescent protein, right? And if you soak the zebrafish embryo in the pool of DNA cage, what we found was, at least, ऐसा नहीं बोल रहा हूँ कि they can absolutely cross the barrier, some molecules of the DNA cage were able to get internalized in the brain of the fish, right? This gave us a potential that maybe, as I'm claim nahi kar rahe hain aaj ki our devices can clearly go to the blood brain barrier, those experiments are still ongoing, but they have enhanced capacity to get internalized even in the tissues like the brain of this. And then once we do that, then what we can do is we can use drugs, active drugs, to induce the neuronal differentiation or neuronal proliferation. How do we do it? उसके लिए technologies आते हैं by which you can induce the stem cells differentiation, right? तो एक molecule आता है जिसको बोलते हैं retinoic acid. Retinoic acid is a very, very important molecule which can trigger the differentiation of the neuronal stem cells into the full neurons. And we said that brain के अंदर तो ये देखना मुश्किल है. Can we replicate it in the petri dish? Can we see that? And what we did is we used the stem cells, and we found that the stem cells can be differentiated into the full neurons. And now what we are also doing is we are going into something called as 3D brain organoid, where you can grow the brain organoids in the petri dish, and then see how these DNA molecules are interacting with them, or how are they inducing the stem cells differentiation. ये 15 साल, पिछले 15 साल का काम अगर मैं समराइज करना चाहूं, हाउ डू वी डू इट, राइट? अभी हमारे सबके पास ये डिवाइस है. वी ऑल हैव समथिंग कॉल्ड एज अ स्मार्टफोन. मेनी ऑफ यू आर राइट नाउ ऑन द स्मार्टफोन, राइट? फोन एक ही है, डिवाइस एक ही है, पर उसके अंदर एप्लीकेशंस बहुत सारे हैं. आप लोगों से बात कर सकते हैं, आप सेमिनार सुन सकते हैं, आप मूवीज देख सकते हैं, आप टिकट्स बुक कर सकते हैं, खाना बुक कर सकते हैं, इवन होमवर्क कर सकते हैं. These are all the applications, the apps which are inbuilt in the device, right? Similarly, what we are doing is we are making DNA based nano apps by which you can have an on demand modification of the protein or the molecules for any question. Then अगर आपको माइक्रोस्कोपी से देखना है, आपको इलेक्ट्रॉन माइक्रोस्कोपी से देखना है, प्रोटियोमिक्स से देखना है, we can do that. Now, जैसा this is where we are today, and where are we going now further? अगर फ्यूचर में आप पूछेंगे कि what is the goal for next 20 years, we are targeting the brain cancers, we are targeting the neurodegenerative disease, we are reaching the sites in the brain where it is getting difficult. So ये हमारा एक perspective था on the applications in the neuroscience, and not only neuroscience, we have to couple it with regeneration, right? चाहे वो body का regeneration हो, organs का या brain का, we are making the DNA cages along with my colleague Professor Sharad Gupta, who is making different peptides. Because remember, the language of the biology is proteins, not the DNA, right? DNA जो है वो सिर्फ carrier है, या उसको आप programmable carrier बना सकते हो. The languages of the proteins. So what we are doing is making a cocktail of DNA and proteins so that they can give signal to the biological systems for future differentiation into 3D organoids, जिसको आज की भाषा में regenerative engineering कहा जाता है और regenerative therapeutics कहा जाता है, right? एक एग्जांपल आपको दिखाता हूँ. ये है umbilical cord के stem cells. We are all born hospitals में ये umbilical cord dispose कर दिया जाता है, but now we are even storing them. So if you grow the umbilical stem cells, ये umbilical cord stem cells on a petri dish, they are circular. ये आपको circular दिखेंगे, right? Now you incubate them with the DNA cages of different concentration, and within 6 to 8 hours, अगर आप नीचे वाला image देखेंगे 400 nanomolar, you will see that they're starting to form a cord-like thing or a tube-like thing. This is the advantage, that using artificial DNA nano devices, we can give instructions to the stem cells for very, very programmable differentiation. And the umbilical cord stem cells were available, so we were able to do that experiments. We are now working with the neuronal stem cells and microglia, and with this what we are now trying to do is we are trying to create 3D organoids, right? एक छोटा सा मूवी आपको दिखाता हूँ how we are going to do that. So ये जो floating cells देख रहे हैं, these are all the stem cells, right? And I can functionalize the surface of the stem cells with the single molecule DNA, And then what we can do is we can capture them on the DNA scaffolds and then grow them in the 3D organoids. So I'll just mute the volume, and यहाँ पे आप देख सकते हैं ये जो है ये living cells है, they are floating in a solution, और नीचे जो है scaffold, that is a DNA based scaffold which will capture the individual cells. Now इसको कैसे करते हैं हम लोग? So what we do is print 3D bioprinting जो मेथड होता है, उससे हम सिंगल स्टैंडर्ड डीएनए जो है वो स्कैफोल्ड के ऊपर प्रिंट करते हैं. ये पेट्री डिश के ऊपर भी आप कर सकते हैं. इट इज वेरी सिंपल टेक्नोलॉजी. यूजिंग अ सिंपल 3D bioprinter you can print the DNA, and this is a single stranded DNA, right? And then हम लोग cells लेते हैं. These are the stem cells or any cells from the blood, and you can pass these cells over this scaffold, and some of the cells they will bind, which have the complementary DNA, they will bind to this scaffold, and the remaining cells will be washed off. Then you can just transfer it on a scaffold, and collagen का जो एक soft matrix होता है वो बनाते हैं, and then we grow them in a very very 3D environment, very closed environment. And once you have this, then you can grow them in the 3D to see how are the different tissues in the body, how do they cause cancer, why are cancers different from the normal body cells, or you can even print the organ like structure, right, in a petri dish. And these will be used for drug discovery programs, drug delivery programs, and then like you can ask many biological questions related to the cancer, and even different 3D organoids, lungs, brain. You can make different kinds of organs on a petri dish. So this was all. This is the future direction that we are moving on. This is my lab here. We are really thankful to all the funding agencies. Thankfully, the lab has been funded. Okay. And like we have very good program of a joint exchange program at Texas University A&M. So our students go to US and Japan, and their students come. So these are the students from Texas who come and spend a semester with us. And जैसा मैंने आपको बताया, being in an IIT एक एडवांटेज होता है हमें कि जो भी डिस्कवरीज हम करते हैं लैब में उसको हम स्टार्टअप में भी कन्वर्ट कर सकते हैं. सो दिस इज आवर स्टार्टअप कंपनी क्यू नैनोसोल, जिसमें हम अलग अलग नैनो मटेरियल्स भी बनाते हैं, डीएनए नैनो टेक्नोलॉजी भी करते हैं, एंड वी आर ओपन फॉर कोलैबोरेशंस. अगर किसी को कुछ इंटर्नशिप के कुछ अपॉर्चुनिटीज चाहिए, पीएचडी पोस्टडॉक के अपॉर्चुनिटीज चाहिए, तो यू आर ऑल मोस्ट वेलकम. एंड दिस इज द ओवरव्यू ऑफ द डीएनए नैनो टेक्नोलॉजी.